Okay, I hope you're keeping warm this day, today, in the next couple of days. Um, Today, we're going to look at a very important section of the immune response. In fact, the lectures that we're going to talk about, the lecture today, the material we're going to talk about today, really forms one of the central cores of the immune response, particularly um, the immune response in which we are going to get antibodies from this response. And so you're going to see these and this information as components of any discussion of the immune response going forward. So this lecture material is pretty critical. So if you do have questions and all, please do make sure you get in touch with me. You email me, that's fine. I'll be happy to answer them. Now, I just want to review that we talked about clonal selection last night, a couple of concepts, and that was clonal selection, and that had to do with the selection, just to respond, that it had to do with the fact that one B cell, each B cell contains, is able to bind to one antigenic determinant, and that antigenic determinant is defined by the antibody that each B cell produces. Each B cell can produce one particular antibody that's able to bind to one particular type of antigenic determinant. You have millions of these. And so that allows us to select, allows the system to select basically for what the B cells that can bind to it, and that allows them, those that are selected can be induced then to go on and proliferate and make more of those cells. And so these are clonally selected and clonally expanded. They proliferate, the expansion being that you can proliferate those cells that have been selected by the interaction with the antigen to the antigenic determinant. Now, these are two really important concepts. It took many years to actually determine this. And these responses is that what is it that, how does this work? I mean, how do you get actually to this clonal through this clonal expansion? And we'll come to that a bit later. Now, the clonal, I guess I've got two of these slides here, mistake. Clonal expansion, once an antibody, a B cell has been selected, the antibody on the surface, the B cell can undergo what's referred to as somatic mutation, in which it will undergo sort of an evolutionary process to make variants of that antibody that can be that will interact much stronger with the antigen determinant, and thus make much stronger binding antibodies that have been evolved to bind the antigen determinant much stronger. And so you can go from a strong interaction to an even stronger interaction. And this is important because it allows the system to evolve, to make your immune system to evolve and make antibodies that have even higher binding affinity and specificity. So if we look at this, what we've discussed, then somatic mutation means once a B cell is found that produces an antibody that binds antigen, the antibodies can be further optimized to increase their ability to bind the antigen. The bottom line, one antibody with a predetermined specificity, which you're born with, predetermined specificity, per B cell, which can then proliferate, generating more antibodies with the same specificity, and through somatic mutation, enhance specificity. Now, if we go back and look at then the immunological response, if you recall, we have the primary response, we have the primary response, we have this primary response, then we have the secondary response. Where do these things fit in in this process, the response? Um, and so what we see is that clonal selection, expansion and differentiation, and somatic mutation and the memory response, somatic mutation, occurs all in this first part. So you're selecting out the cells, you select out the B cells that are important, they are expanded. We'll see they will differentiate into cells called plasma cells, which are going to produce antibody. I'll have more to say about that later. And then through this process, you also see somatic mutation, and most importantly, the first time I've said this, there's a memory response in which there are memory B cells that are produced. Memory B cells. These are long-lived. And so all of these follow this process, and then this comprises what you would see in the primary response. Now, upon secondary antigen, if we have a second antigen insult dosing, basically, because there's a memory response, what you have memory response, then you're going to see a much higher, steeper interaction, with the immune system, producing more antibody and much more quickly. And this is quicker because there is memory here, because you've got a memory. You're engaging memory cells here that who remember interacting with that the past antigen. And again, you can have additional somatic mutations that can occur in the secondary response, creating even higher affinity antibodies. So each time, potentially higher affinity antibodies. And so as you can see, clonal selection, expansion, and somatic mutation are part of that primary response. And then upon a secondary response, you make use of those the memory B cell response, which allows for a greater increase, faster response to occur upon exposure to the antigen, and then further optimization. So the central characteristics of this are B cells are each programmed to respond to an antigenic determinant by proliferation and differentiation to produce an antibody against the antigenic determinant. So the B cells are these central players. They have a pre-programmed disposition for that. The initial specificity is predetermined. B cells have cell surface receptors, and these are the antibodies that we've seen. They're called B cell receptors, antibodies that are bound to the surface. The binding of the specific antigen triggers this response, so the specific antigen interaction is important, so your specificity there. And due to somatic mutations, the IgG produced by later responses is also characterized by an increased affinity. So those are all sorts of key principles or essential characteristics you'll find in this response. Now, how do we generate all of these different antibodies, a generation of antibody diversity? There are literally billions of possible antibody specificities that you can generate, and therefore there must be billions of antibody mRNAs to be able to do that. In other words, what I mean by this is that you have the ability to produce many billions of different types of specificity, but that antibody, in order to produce that, the genome would have to be almost entirely made up of DNA that encoded antibodies, and that it does not. And so how does this happen? The way it happens is to generate this level of different types of specific antibodies on the order of the scale is by the use of recombinations and somatic mutations. In the somatic mutation, which we've just seen, but also recombinations can enhance tremendously the diversity that is generated. So I'm going to give you just a brief primer on this. We could go into a lot of detail about this, but I just want you to be just familiar with this. And that is, if we were to look at the light chains, right? The light chain, for example, if we look at this light chain, the kappa chain, for example, there are several segments of this. There are genes for the variable region. There are genes for the joining region that joins the variable region to the constant region. There's less diversity in the constant region. And so there's a number of different combinations. If we were to multiply all of this together, we would get 7,500 different combinations. And so there's a number, most of that variation being occurring with the variable region here. Now there are diversities within the constant regions because there's also light heavy chain diversity as well. And so they have to fit together. And so they will be selected based on that as well. There are also, for the heavy chain, there are over 80 regions that are variable regions. The joining region contains at least 100 genes, and there may be 300 different constant regions that you see for an antibody. And so if we look at this, the flexibility, there's possible like 2.5 million combinations that we can have here of the different types of antibodies that we can potentially produce, of different types of heavy chains that we can produce. If we take those and we look at the total diversity of this, if we just multiply those then together, 7,500 times 2.4 million, we get 1.8 times 10 to the 10th different combinations. And that alone gives you a tremendous potential diversity across the different antibodies that are potentially available. In addition to this, of course, are different mutations that can occur and different recombinations that occur within the variable regions. And so these genes I have here that are 150 genes and 80 genes, these would be just the very first genes, but these can be recombinantly, you can undergo recombinations as well to produce a whole range of different genes in the variable regions and in both variable regions, which we can't really account for here, which produces another set of millions of different potential different antibodies. Suffice it to say that that's because of these ability to recombine the DNA within the B cells and create molecular changes, mutations within the B cells, that the different types of B cells can be generated. Now, the diversity that you actually have probably about 10 to the 8th different any one time, about 10 to the 8th B cells at any one time. And so the diversity there, the diversity that's possible is way above the diversity. In this case, 10 to 10th, maybe 10 to the 12th, different potential antibodies. So the number of B cells that you can produce is well below the potential diversity. This is important because in any one time you have no idea what antigenic determinant or antigen you're going to run into from a pathogen, and so this allows for basically an overabundance of diversity within the system, so that you're constantly throughout your lifetime you can produce antibodies with different potential specificities throughout your lifetime. Now, having said all that, that's all I'm going to say about that. We could spend like a couple of months talking about how antibody diversity comes about. I just want you to know that there's billions that can be made, that it goes through, again, recombination events and mutation events to produce those. The mechanism, though, by which antibodies are produced or excreted to an antigen is referred to, and this is the next section. These next sections are really critical. Is referred to as T-dependent AMI. The reason for this is it is dependent on T cells, and we're going to get into that, where that T comes from. T-dependent AMI, immunity has evolved to produce strong, specific responses of long duration that will protect against diseases most frequently encountered by soluble proteins and cell surface antigens. So soluble proteins and cell surface antigens. And so later on we'll talk about how the immune system protects against viral infections and cancer. But when we're looking at soluble proteins and cell surface antigens, the T-dependent AMI is critical. It's characterized by different primary and secondary immune responses, which we've seen. IgM is initially produced, then IgG or IgA, depending on where the response is. And you can have increased affinity with time, as we've seen in the primary and the secondary responses. Now the players. There are several players to this, to the AMI. One of the first we've seen are macrophages. These are large phagocytic cells that are referred to as antigen processing or presenting cells. And so these antigen presenting cells and processing cells are called APCs. And these are cells, as we'll see, that are going to have a particular function in interacting with T cells. These APCs, like macrophages, have no antigenic specificity prior to immunization. What that means is that they don't carry any antibody. They don't carry any. They are agnostic with regard to what the antigen is that they're going to interact with. They do express two molecules, which we're going to go in detail with. One of these in detail shortly. The major histocompatibility complex 1 and major histocompatibility complex 2. We're going to talk about that later, so don't worry about that. And it also produces another chemical factors in response to its interaction with the antigen. And we've seen what this is, again, the antigen. This is a macrophage, and the macrophage is chasing this bacteria, as you can see, and it's when it eventually engulfs it. As you can see, it's going to tag it. It's tagged it. Oh, sorry, it went backwards. Sorry about that. When it tags it, engulfs it, that's when the antigen processing presenting is going to occur. Now, another set of cells that's really important are these dendritic cells. And these are professional antigen presenting cells, the dendritic cells. They have no antigen specificity prior to immunization like macrophages. They also express MHC1, MHC2, and they produce also a number of chemical factors. And they are found in the peripheral tissues, lymph nodes, and spleen. These are secondary organs of the immune response. And later on, we'll talk about where they are. Macrophages, I just want to point out for macrophages themselves are found throughout the body, and they have multiple functions, like, for example, clearing you of cell debris as well as antigen presentation. But dendritic cells are actually professional molecules in which their only purpose really is to interact with the immune cells and interact as well with T cells. They get this name of being dendritic by the fact that they have these dendrites, these long spidery protrusions from their membrane, as you can see here. They're like a dendrite. That's where you get the name dendrite, and therefore their surface area is quite high, enabling them to interact with multiple cells simultaneously. If we look at this, a second here. Hold on a second. If we can see one here, you can see here in this little movie, you can see this dendritic cell, and you can see these little protrusions that are just it's always sampling its environment, and there's a whole bunch of them, and they're constantly kind of on the move. They move around and in search of cells to interact with. And as we can see here, it's coming up. they really want to interact with specific types of cells, and those specific type of cells um, um, are T cells. Let's see here. It's coming up here. Yeah. So here you can see here are these dendritic cells, and then these other cells, these little cells here, are then T cells that it's interacting with, and they can interact with multiples of these cells simultaneously. And you can see that here. One cell. This is really good. This is a dendritic cell, and you can see it's like dragging along a whole bunch of these T cells and interacting with them, and causing potentially activation, as we'll see. It's really cool. So the other cell that we've already talked about, are B cells, and these are the ones that are produce immunoglobulins. They can be considered APCs, and they also express MHC1 and MHC2. So they again do this antigen processing. Upon activation, they can divide to produce plasmacytes, and these plasmacytes will produce antibodies, and they also produce memory cells, so that there's some record of the interaction that they've had with an antigen. They bear several distinct markers, and these markers are, as you'll see in immunology, are markers on the surface are referred to as CD, or cluster of differentiation. That's cluster of differentiation, and they're given a number, and there's actually an organization that does this when a new factor or a new surface marker has been identified. And so we can identify B cells or by these particular markers here on their surface, proteins on their surface, as well, and we can identify other types of immune cells by different types of different types of CD molecules on their surface. And you can see here's a B cell. They're round. They have a very fluffy outer membrane, and again they express antibody on their surface. Now the real major player that we haven't talked about are T cells, and there's a particular type of T cells that are necessary for the antibody-mediated immunity, and those type of T cells are referred to as T helper cells, and they promote the optimum immune response, and in fact they're central to the whole, as you're going to see, central to that immune response. They bear distinct surface antigens that are not on B cells but are only on T cells, on these type of T cells, and those are referred to as CD4 and CD28. These are two types of molecules that you'll find on their surface, as well as a T cell receptor, and we'll have more to say about that later. They also have a CD3 complex, which is necessary for their activation, as we'll see. They can be readily cultured and cloned using mitogens. So these are activators. These are ones that cause mitosis. They and activate the cells to divide. And they also produce powerful chemical signaling molecules. The most important thing, one of the things I do want to make sure you understand, is that these molecules, helper cells, these T helper cells, are also referred to as CD4 positive cells. So these are CD4 positive T cells, and you'll have more to see about that in a couple slides. There are different types of T cells, as we're going to find out, and the first type of T cell that we're going to focus on are these CD4 positive cells. Now, the mechanism, how does AMI work? Well, if we have an antigen, and this is an antigenic determinant right here, there's a couple antigen determinants on the antigen. That antigen can interact with a macrophage or dendritic cell, and they will be taken up. There are several, you've seen the PAMs, the different molecular recognitions possibilities that they have. They'll phagocytose They phagocytose the antigens by the mechanisms that we've talked about. And by these mechanisms, the antigen is internalized. The internalization process means that they form the endosomes and within that process, there are in the endosomes that they are sequestered in the cell from. When this happens, this process induces them to produce. They produce actually B7 on their surface. This is a molecule that we'll see later is important in interacting with T cells and signaling with T cells. This internalization by the macrophage or dendritic cells undergoes now a process in which the antigen itself, the antigen is digested into a series of peptides. And what I have here is these are peptides. These little balls here, black and red, are which we've cut up basically by proteolyzed it. The antigen has been proteolyzed into pieces or peptides. Now these peptides are important because these peptides, this antigen presenting process we're going to talk about now has some complexity to it because these peptides come from the antigen and they're important and they're referred to as we'll see as antigenic peptides. Now what happens when you get this this endocytosis of the antigen? So let's just say we have an antigen, it is endocytosed. There's, for example, antibody on the surface or a PAM that can bind to that. There may be other receptors on here. I've just used this as just a type of receptor that will endocytose, that will result in the endocytosis of the antigen. We'll see later that antibodies can bind to macrophages and dendritic cells. They have receptors that will initiate this, but not in the beginning. What happens is that you get endocytosis of the antigen, and this endocytosis results into hydrolysis into those peptides. Now there's another part of the cell referred to as the rough endoplasmic reticulum, in which this molecule MHC2 has been expressed, and it's expressed with what's referred to as an invariant chain. And this molecule contains two chains, and we'll go into detail about this, two chains. And this invariant chain here in blue, so this invariant chain's in blue, binds into a binding pocket that we're going to see binds to a peptide later. Now this will move, this will be moved in the process. There's sort of a constant, this is sort of constantly going on, in which we have digestion and removal of basically this invariant chain to produce basically a much smaller, referred to as clip clip in the MHC2. This is sort of a placeholder. It's a placeholder, basically peptide that's lightly bound into that surface. Now that chain, that the two membrane bound the endosome and this MHC2 clip endosome then will come together and fuse, and as a consequence the peptide is going to come into contact with the MHC2. This results in, if this peptide is able to bind, if there's a peptide in here that's able to bind to the MHC2 complex, then it will displace the blue clip peptide from its surface. And as a consequence of this exchange, what you get is you get a peptide that's from the antigen that's now bound to MHC2. So this is called clip peptide exchange. Once this occurs, that MHC2 is then targeted and transported via the Golgi to the membrane and displayed on the surface. And so what we see here is a peptide here, this peptide in red, that's from the original antigen, it's from this original antigen over here, is now displayed on the surface of the cell, of the dendritic cell or macrophage. And so there may be, I want to point out there may be multiple copies of this because you take up several different antigens, but these will be displayed on the surface of the macrophage or dendritic cell. So you go through MHC2 fusing with this endosome, and then exchange of the peptide from the antigen, exchanging the clip out, and then transport of the MHC2 peptide to that surface. Now the macrophage or dendritic cell is going to produce, it's going to display the peptide on its surface, and there, as we'll see later, you have a variety of different MHC2s, and so you can produce, a macrophage could potentially display, for example, there's a black one here peptide, there's a gray peptide here, different peptides on its surface, as well as more of the different peptide as it phagocytoses more antigen on its surface. So there'll be many of these that'll be potentially produced on the surface of the macrophage or the dendritic cell. Now, in the initial stages, there's an interaction then between the MHC2, and I'm sorry, that should be MHC2 here. Let's see, 2, okay. So MHC2 and the peptide and a molecule called the T cell receptor. This set of interactions, as we'll see, is really critical. This is a really key set of interactions, and because each T cell, as we saw with B cells, they produce a particular antibody or B cell receptor. Each T cell produces its own specific type of T cell receptor that will recognize a peptide and the MHC2. It also has in this cluster, this complex, a molecule called CD4, which we've seen before, and that molecule functions as a clip, a molecular clip to hold this complex, the T cell receptor, in place. It recognizes the MHC2. There's another molecule called CD3, and this is a molecule that results in signaling that helps in signaling to the T cell, which is going to be necessary for its function. Now, remember I talked about the B7 is on the surface of the macrophage and dendritic cells, and is expressed when they undergo this antigen processing. You get more expression of B7 on the antigen processing cells. They bind to another molecule which you'll find only on T cells, and that is CD28. So they form this complex, the B7 CD28 complex. This is really important because it's referred to as a secondary signal. A secondary, you have a first signal and then a second signal to the cell, to the T cell, for activation. So you need both activation, as we'll see. We need both signals to get activation. Now let's take a little closer look at the MHC2 complex and how it's constructed. The major histopathology complex is composed of two polypeptides. We can see here, there's a polypeptide, the alpha chain, and then the beta chain, the alpha and beta chain. And they are similar in size. They're roughly about 30 kilodaltons in size, but they're not the same. There are two different gene products. Each one has a domain alpha 1 and alpha 2, and the beta chain has beta 1 and beta 2. These are two domains, two additional domains, 1 and 2, which are defined in the MHC2. And they also have a cell membrane spanning region, and then they also have a cytoplasmic region, which is spans and sticks into the cytoplasm in both cases. So the MHC2, actually, you need to have these two chains. There are regions, these are carbohydrate, potential carbohydrate binding sites. And so those are, and they differ on the MHC2s. But for what we're going to talk about, they're really not that, don't have that much function. Now these domains, alpha 1, alpha 2, beta 1, and beta 2, also have disulfide bonds in them. And these disulfide bonds help to stabilize that the secondary structure within them. Now, if we look at this, so here's a movie of one. So if we see there are two chains, the alpha and the beta chain. Here, one in red, alpha in red, the beta in blue. And if we can see, I'll stop there. You can see what happens is that when they come together, they form a pocket. And that pocket can bind a peptide. And as we'll see, it can bind both versions. Both are necessary. Both domains are necessary. And both domains are necessary, and both chains are necessary to form this pocket. We can see here kind of nicely that the chain there's a part that is surface exposed, solvent exposed, and there's a part that's bound, that binds into this groove. Take another look at this. So we'll just continue this around just to show you. And you can see they composed of these alpha chains and beta chains. And those chains are made up of secondary structure is, again, beta sheet, which is quite stable. But interestingly enough, and we'll come back to this, the chain itself, one of the parts you'll see is an alpha helix in the top, which comprises basically the top of the chain. Now, if we look, and you can see here the peptide binds across this binding site. I'll let this go a little bit longer, so you can see it for MHC2. This peptide's binding across this whole site, and we'll just let that go. Okay. Now, if we look down on the chain, if we look down, if we were to look down on the MHC2 peptide complex, what we'd see is that the binding site is composed, as you can see here, composed of both. You get blue and red composed of both chains, the alpha and the beta chain. They both contribute alpha helical sites that are needed for binding. As you can see here, there's a blue alpha helix and a red alpha helix. And then half of the binding site for the peptide comes from one chain. You can see here from the red, and half of it comes from the other side. And you can see the floor, basically the floor of this of the MHC2 binding site is composed of both chains. And then the peptide sits almost like a hot dog in a bun on the top within that binding site. Now, if we look at take a look at the interaction of the peptide MHC2 with the T cell receptor, what you can see is again what we see is the peptide is bound, and there's a surface exposed part of this. So there's the peptide here in the middle in the MHC2, and it's presenting. That's why it's referred to as antigen presentation. It's presenting this peptide to the T cell receptor. Now look at the T cell receptor. It looks kind of familiar, doesn't it? It has these beta sheets, and guess what? It has loops that stick down into it and to recognize that peptide. So it's actually a member of the family, the immunoglobulin family, that similar to antibodies, and so they actually have a lot of similarity to antibodies in that they have loops. There's two chains, which again as you can see here, actually generate 3 loops. So there's a brown, there's 3 loops. The yellow has 3 loops. And then you can see the loops in the middle, the third loop, as we said for antibody binding. You can see here and you can see the loops interacting directly with the peptide. And so these central loops, as well, I'll just let this go here, stop it here. Dang it. Second. Okay. We look here, we can see this loop, this yellow loop is touching the peptide. And so it's touching that peptide. Now, it's maybe not quite as obvious here, but those two loops are touching that peptide and directly interacting with that peptide. This part of the peptide, of course, is not binding, is solvent exposed. The other loops can bind also to the peptide, but they also interact with, so let me just show you here, stop it here. If we look here, you can see this loop here interacting with the actual MHC2. And so this points out a really important thing, is that the T-cell receptor not only binds and recognizes the peptide, the molecular recognition of the peptide, but it also recognizes the MHC2. So the MHC2, as we'll see later, there are many variants of the MHC2 because there are many different types of peptides. It needs to be able to bind to many different types of peptides. And as a consequence, there's also many variants of T-cell receptors, and they look to bind not just the peptide, but also the MHC2. And so it's a double recognition in which the T-cell receptor binds to the peptide, and it also binds to the MHC2. Conversely, the MHC2 interacts directly with the T-cell receptor, the alpha helix interacting with this loop, and through peptide binding, it interacts directly with the loops of the T-cell receptor. So it forms basically a little sandwich of T-cell receptor, peptide, and MHC2. And all of this molecular recognition is critical to going forward with activating the immune response. If it doesn't exist, if this interaction is too weak, isn't recognized, then no response will happen going forward. Now, we talked about T-cell, the CD4, and this is a molecular structure. It shows you the interaction of the MHC2. There's a peptide up here, this little wiggly guy, and it binds the CD4 now binds to basically the lower part of the MHC2 that doesn't bind to, and you can sort of see that here. The CD4 binds to the lower part of the MHC2 that's not involved in antigen binding, antigenic peptide binding. So the CD4 binds to a lower part and recognizes in a very conserved fashion their sequences down here. These domains down here are highly conserved, and so it binds in a conserved fashion to CD4. There are not multiple types of CD4. There's only one type, and then these domains are constant across those across different MHC2s. It's only up here that only in the top part of the molecule that you're going to see variation in sequence. And so you can see here there's pretty tight binding, and so if you have the CD4 bound as well, then that further stabilizes that interaction with the T-cell receptor, peptide, and MHC2. So if we look here, here's MHC2, just to sort of show you again in this diagram. If that wasn't clear enough, the alpha chain and beta chain contribute equally to the peptide binding groove, both at the floor, this beta sheet floor, the floor of the MHC2 binding site has beta sheet structure, and then on the outer surface binding, sort of almost like teeth, as you can see here, is the are these two long alpha helices. So if we look at T-independent AMI going forward, we have these set of interactions. There's a set of very specific interactions with the MHC2 and T cell receptor here and the CD4. And so the next event is that in this process, if IL-2 is referred to as interleukin-2, it's a signaling molecule. It's a peptide, protein peptide that is a cytokine, referred to as a cytokine, and is produced by macrophages and dendritic cells. Then what happens is this Th0 cell, referred to as a naive. A Th0 cell is a cell that it's a T cell, a T helper cell that has not interacted with a macrophage or dendritic cell before. But now that it has, it's sort of lost its virginity and is now a Th1 cell. And if it's in the presence of IL-12, now it's a committed IL-2 Th1 cell. If it's in the presence of IL-4, which is produced by Th0 cells and Th2 cells, which might be in the vicinity of this interaction, then another type of cell is produced, and that's Th2, a T helper cell, CD4 Th2 cell. So you can see there's two different types of these CD4 positive cells. Each one is identified as CD4, but they have different. That we'll see they're going to have different functions, Th1 and Th2. Th2 cells are critical because the Th2 cells are the ones that are necessary for antibody production and the increased antibody production, as well as B cell proliferation. And they can produce IL-4, IL-5, and IL-13. So these are molecules that are activators, chemical signals, peptide signals that can be produced by them and are important in the next steps of the process. So you now have a Th2 cell, and that Th2 cell in this process can divide when it's activated into a memory cell, a Th2 memory cell. It can also begin to seek out activated B cells, and so it can divide, and then the Th2 cell itself, which is an activated Th2 cell, can seek out seek out B cells to interact with. And so here you have, for example, here's a B cell, here's a T cell, and this is a antigen processing cell. And you can see it travels, it now interacts with the membrane, it's interacting with the membrane, and it's undergoing, in this process, it undergoes series, as you can see, it's really sampling that membrane, finding what the specific interactions are, and we're going to see in detail of that. And then what it does is it's going to leave the antigen processing cells and seek out to interact with a potential B cell. And so the antigen can now, you have basically a prime system. And now remember we talked about a B cell interacting directly with the antigen through antigenic determinant. Well, antigens interact with the antigenic, basically through the antigenic determinant through B cells that will express largely IgM. IgG is not really that important, but IgM is on their surface. They'll produce with that specific specificity, and they're referred to as naive B cells. They've never had an interaction with a T cell before. And so they can form either single, or if there's, in this case, I have this antigen has two of the same antigenic determinants on it, or it can bind to the antigen antibody and maybe cross-link those antibodies. Regardless, what it does is it can undergo, then the antibody will be internalized with the bound antigen, will be internalized into the cell. B7 will be expressed on the surface of the B cell. That antigen will start to undergo, that internalization of that antigen will start to undergo then antigen digestion to the peptides, as we've seen before. And this is all the same as what we've seen with the macrophage and dendritic cells. So you already know this part. There's another molecule that's produced on B cells, and it's called CD40, and we'll come back to that in a minute. That's important later on. Now, this antigen digestion of the peptides results in then MHC2 peptide presentation, as we saw through the same mechanisms that we saw through the macrophage and the dendritic cells. And they too can produce an MHC2 peptide, potentially different peptides can bind to different MHC2s in the cells complex, and then those are presented on the surface as we can go through this whole process again and produce more B7 and CD40 on their surface. So this whole process we've seen before. So this is why B cells are considered antigen processing cells as well. Now we've seen this before. The naive B cell, now that's producing an MHC2 peptide on its surface, can now interact with the T cell receptor in the same kind of process. The T cell receptor interaction now is going to be mainly through this MHC2, through these MHC2 TH2 cells that were generated before with the dendritic cells and the macrophage. Then the TH2 cell now, when it runs into a B cell that's expressing the peptide MHC2 that it recognizes on its surface, it will engage it. Its CD28 will engage with the B7, and CD40 ligand will engage with the CD40. And so you see this whole complex will now be formed. The only difference that we've seen with the macrophages is that there's this CD40 ligand interaction with the B cells. I just want to point this out that on B cells, to specify that interaction on B cells is what I'm interested in focusing on. The TH2 cells, these TH2 cells, when they do this, can be activated. They'll form more memory cells, divide, and they'll also form more, let me go back here. They'll also divide to form more TH2, to form more memory cells, as well as more TH2 cells. And so this interaction also results in the activation of the B cell. And so this naive cell now becomes an activated B cell, in which it's encouraged to produce more antibody on its surface, B7 and CD40. And this means that this activated B cell now also divides, and so we've been clonally expanding it. So remember the clonal expansion? You're clonally expanding these B cells. You're getting, when your cells activated, they will divide and proliferate. And so we've clonally activated the T cells and we've clonally activated the B cells. Now the activated B cell can go on to form more B cells, form memory B cells, and form more copies of the activated of itself. It can also differentiate. It will also differentiate into plasmacytes, and these plasmacytes will produce the antibodies that are initially they may produce some IgM, but they quickly produce IgG and IgM, and if it's an allergen, it would be IgE. But for what we're concerned with right now, we'll come back to IgE in the hypersensitivity stuff, hypersensitivity lectures. But it's going to produce large. These are the ones. These plasmacytes are the producers of the antibody, and so the B cells need to differentiate into these sort of hyper B cells that are really antibody factories that will excrete the antibody that's necessary. And of course necessary to produce memory B cells. Now the activated B cell could interact with an antigen again, and that antigen can, once it's interacted with the activated B cell, it can go out and potentially interact with a naive cell Mainly it interacts with these additional Th2 cells and producing more Th2 cells, activated Th2 cells, more Th2 memory cells that you have more of the same. They're being more, this interaction leads to proliferation and production of differentiation into Th2 memory cells as well as additional Th2 cells, activated cells, more activated B cells, more plasmacytes, and more memory cells. So you get this expansion of the whole system in which B cells, activated B cells are expanded, the amount of memory cells are expanded, the more plasmacytes are expanded. What I mean by expanded is more of those cells are being produced. You get the expansion of the T cells in which you get more of those T cells that are necessary, more and more T cell, more T helper cell memory cells. So the key thing here is upon this response, you get activators that can give more activation to boost the amount of antibody that's being produced, and the system begins to learn and begins to register and to register the interaction it's already had and produce cells that will keep the memory of that interaction long term. Now, let's just take a look. This is a little bit of a review. But I want to make a point about this because there's a few other molecular players that are involved and between the cells that we've talked about. If we look at a cell, the cell interaction, this is the membranes between the two cells. There are these molecules that are really critical to interacting and holding the interaction between the cells together. These are referred to as cell adhesion molecules. And for a B cell will produce LFA2/3 that interacts with CD2 on a T cell. ICAM1 is produced on B cells, and LFA1 is produced on T cells. And these form specific interactions. They're like molecular Velcro on the cells to kind of stabilize that interaction. We then, of course, have the what we've talked about, the T cell receptor MHC2 peptide complex is formed, the B7, CD28 secondary complexes form in the B cells. And then you also have what's critically important is the CD40, CD4 ligand interaction between the T cell and the B cell, because this is important for class switching. This interaction enables long-term the switching of these antibodies to from IgM producing to IgG, as we've seen in that primary response. Now okay, I think I got 2 copies here. What I failed to mention, or I just want to produce, is that the activated T cells are going to produce these cytokines, IL-4, IL-5, and IL-13, and these help to boost the activation of the T cells. It boosts the class switching. It further activates class switching and proliferation of the cells, and so these are really important as well, and we'll see that, and you'll see that. Now for a Th1 cell, I just want to point out. Remember, we get Th2 and potentially Th1 cells. We're going to come back to this, but I just wanted to point out that these Th1 cells are important because they enhance, they will drive potentially B cell, T cell proliferation and macrophage activation by the production of IL-2, interferon gamma, and TNF beta. And so these particular these 3 here are really important because these cells, these T cells produce these molecules here, IL-2, interferon gamma, and TNF beta, are really strong activators of the immune response, and they'll activate particularly macrophages and B cells. And so when we see the APC or macrophages interacting, they'll interact with these Th0 cells as we see in your Th1 cells. They have the same cell adhesion processes, interactions. Again, the T cell receptor MHC2 peptide complex is formed. B7, CD28 is formed. But in this case, what I want to point out is that these guys can produce these CD28 will produce IL-2. And IL-2, the CD28, we're going to look at that later, but this interaction results in a strong activation of the production of this cytokine IL-2. This IL-2 gets excreted and will further activate, these T cells and causing them to divide, as well. I guess I got two parts of this. I didn't realize that's the case. Oh, and one of the side effects or the responses of this IL-2 generation of IL-2, and then the response of IL-2, basically IL-2, this paracrine response in which the IL-2 receptor is produced by the T cell and then takes up its own IL-2 in this continuous activation, is again the production of interferon gamma and TNF beta, which go a long way to activating these antigen processing cells, resulting in antigen processing activation. Now, what I've done here, I know this looks scary, but what I've done is try to summarize it, put all the pieces together for you so you can kind of see how the whole thing fits together. So if we go up here at the top here, so it's not as scary, and actually, I know it's a lot of information, but if you start looking at it, you can start taking it apart, and you can see that there's a lot of common things here, commonality here. So if we have an antigen, if we have this antigen, it interacts with the macrophage and dendritic cell, you get antigen processing. In the presence of IL-12, you get Th1 cells produced. IL-4, you get Th2 cells are produced and proliferate. You also get memory cells that are proliferating. These Th2 cells can go and interact with a B cell. The B cell, a naive B cell, then that has interacted with the antigen, it's producing a certain antibody, and then this enables then the Th2 cell and the B cell then form this complex with this naive B cell, which now leads to activation of the B cell, and the B cell activation leads then to plasmacytes production, memory B cells being produced, further interaction with the antigen. B cell would interacts with the antigen, leads to additional antigen processing, additional T cell activation, more T cell activation, and more memory cells, plasmacytes, activated cells, Th2 memory cells, and Th2 cells. And so what you see here is that there's just a general proliferation from initial events all the way to production of three really important things: plasmacytes, activated and memory B cells, and activated T cells, T helper cells, Th2 cells, and Th memory cells. So I think that's actually 1, 2, 3, 4, 5 things. But you actually get cells that are produced by those products of this reaction. B cells and different types of B cells and T cells that are necessary, and antibody. And that's where antibody comes from. And that's how this is all what's going on in that beginning, that latent phase. And as you see the antibody go up in the plasma, that's what's happened. All of these events have happened. It takes time for that to happen. That's why it's a latency. It takes time for all of this to occur. And so that's why you don't have an immediate production of antibody, but you have to go through and have all of these interactions occur. There's specificity in these interactions. Remember the MHC2 peptide and T cell receptor interactions best specify the interaction with that antigen. And then the other part is the antibody direct interaction with through the antigen term through the binding to the antibody. So those are two critical things, and if those specificities aren't there, if you don't have a good MHC2 peptide T cell receptor interaction, then this won't happen, go any further. If you don't have an antibody binding to the antigen, that interaction is too weak, then that's not going to happen, and none of this will happen. Okay, so I'm going to stop there. I know that was it's a lot to digest, and we're going to stop there. And I'll come back in the next talk. I'll talk a little bit more about the interactions between the T cells, what we know about that, and yet another different mechanism that is in which antibodies can be produced that's simpler and more archaic.